Welcome to this explainer. When we usually think of a virus, we just picture this biological pathogen, right? Something that just makes us sick. But today, I want us to totally flip that perspective. We are going to look at the virus as a highly efficient, hyper-coordinated microscopic manufacturing plant. From sourcing raw materials to assembling complex machinery and then shipping out the final product, the virus is honestly an absolute masterclass in automated production. Let's get right into it. Here is our factory floor plan for today. First, spying on viral factories. Then, the microscopic assembly line, packaging the genetic code, acquiring an envelope, and finally, escaping the host cell.
All right, let's start with section 1, spying on viral factories. So before we can even begin to understand this machinery, we have to know what kind of incredible investigative tools scientists are actually using to infiltrate and observe these tiny, hidden plants. Our heavy hitters here are two entirely different types of electron microscopes. We've got the scanning electron microscope, or SEM, which fires electrons to scan the outside surface of our sample. And then, on the flip side, we have the transmission electron microscope, or TEM. This one literally transmits electrons straight through the sample. These two tools give us fundamentally different perspectives of the exact same viral particles.
Now, just look at this striking visual difference. On the left, the SEM gives us this incredible, super colorful, 3D surface-level view of the viral particles all clustered together. But then on the right, the TEM gives us the internal blueprint. You can clearly see the dark, dense cores of the viruses and their internal structures. It's basically the difference between looking at the outside of a brick-and-mortar factory building and then looking at the X-ray floor plan of what is actually going on inside. But, static images really only tell us part of the story. If we want to see the factory actually in motion, scientists had to turn to nature, specifically to this fascinating creature, the Aquaria victoria jellyfish. It naturally glows green in the dark, and believe it or not, it provides a totally crucial tool that lets us track these tiny viral parts live, like in real time, right there on the assembly line. How do they do that? Well, they extract the exact DNA sequence that makes the jellyfish glow, and they attach it to a viral protein. This creates what's called a fusion protein, specifically using green fluorescent protein, or GFP for short. By using this as a super bright biological tag, we can literally watch the virus components moving through the host cell. We can track them live through the entire infectious cycle without having to just guess where they're going. It's a real game changer.
Moving right along to section 2, the microscopic assembly line.
Okay, this is where the whole idea of our viral cell operating exactly like a coordinated factory really comes to life. Picture a highly automated car manufacturing plant. You've got robotic arms actively welding and assembling cars, right? Well, a virus works exactly like this. Viral assembly is absolutely not random. It uses a very specific parts list to form discrete sub-assemblies efficiently and on a highly orderly time frame. Just like you wouldn't build a car by just throwing all the metal into a pile and hoping it drives, viruses build distinct parts first before putting the whole shebang together. And to do this, the viral assembly line relies on 3 distinct manufacturing strategies to create these subassemblies. First, they can assemble them straight from individual protein molecules. Second, they can carve them out from a much larger polyprotein precursor. And third, they use chaperone-assisted assembly, which is where helper proteins literally come in to assist in the complex folding and construction process. It's so cool to see exactly how these complex sub-assemblies physically fold and snap together. You can see how the simian virus 40 forms from individual pieces that just snap right into a pentamer. Then, look at the poliovirus. It uses a long polyprotein chain that gets cleaved or cut into structural units. And finally, you've got the adenovirus type 2 relying on that chaperone protein right there in gray to help build its complex hexon trimer. It is literally microscopic engineering at its absolute finest.
That brings us to section 3, packaging the genetic code.
So our virus factory has built its protective shell, but now it faces a massive mechanical challenge. How on earth does it manage to stuff all those delicate genetic instructions into that newly manufactured container? There are 2 main packing methods for this, known as encapsidation. First up is sequential encapsidation. This is exactly what it sounds like. The sequence is to build the empty protein shell first and then insert the genome into it later. Poliovirus does this. The stark contrast to that is concerted encapsidation, where the viral particle and the genome assemble together simultaneously at the exact same time and place. Influenza is actually a really great example of this all-at-once method.
Let's zoom in on the specific mechanical steps of that first method, sequential encapsidation, using the herpes simplex virus as our model. Step 1, a specialized complex interacts with a literal portal on the empty shell. Step 2, the DNA is aggressively reeled right into that portal. Step 3, exactly one full genome is packaged inside. And Step 4, specialized PAC sequences act like tiny scissors to cleave or cut the DNA once the shell is completely full. And when you visualize this process, you quickly realize this is not passive. The virus literally builds a mechanical motor that latches onto the DNA and actively pumps and reels it into the incredibly tight geometric space of the capsid. It's a super high-pressure packing job that requires immense microscopic force. But wait, what exactly is the motor pulling in? Well, this introduces a genuinely fascinating concept called a concatemer. Instead of making single copies of its genome one by one, the virus uses continuous DNA synthesis. So it creates this massive, long, repeating chain of genetic material, just waiting to be processed and snipped into exact genome-length pieces by those PAC sequence scissors we just talked about. I absolutely love this comparison because it brilliantly illustrates the idea. Think of a concatemer like pulling continuous sheets from a toilet paper roll. The DNA synthesis just rolls out one long continuous strand, and the viral packaging motor keeps pulling it in, snapping off exactly what's needed for one single virus and then moving right on to the next one. It is just so beautifully efficient.
Let's move on to Section 4, acquiring an envelope.
You see, for a lot of viruses, a simple protein shell just isn't enough. They need an outer lipid envelope to actually survive and infect. They acquire this through a process called budding at the plasma membrane of the host cell, which basically makes this the specialized packaging department of our biological factory. But not all budding is the same. There are 4 unique types, and they're driven by completely different proteins. Type 1, like the alpha virus, uses envelope glycoproteins and the capsid. Type 2, like retroviruses, uses the GAG polyprotein. Type 3, like coronaviruses, is driven by envelope proteins. And finally, type 4, like the rhabdovirus, relies heavily on matrix proteins. Just notice how these budding structures literally push out of the host cell's membrane. You can see how the different internal viral components, whether that's a tight nucleocapsid in type 1 or the distinct shapes in types 2, 3, and 4, actually dictate how the membrane wraps around the virus to finally secure that protective outer envelope. And finally, Section 5, escaping the host cell. So we've built the parts, we've packed the DNA, and we've wrapped it all up in an envelope. Now the finished packaged product has to figure out how to ship out and start the cycle all over again. Let's look at the highly mechanical ESCRT-dependent release process, which is used by retroviruses like HIV. Step 1, the JAG proteins group up and start bending the membrane outward. Step 2, the host cell's own ESCRT3 proteins assemble in these concentric spirals right around the base. Step 3, that spiral physically constricts the neck of the budding vesicle. And Step 4, an enzyme called VSP4-ATPase comes in, disassembles the machinery, and safely releases the fully formed virus. The really crucial point is right here in the middle of the process. Notice how the SCARE3 spiral acts almost like a biological lasso? The virus is essentially hijacking the host cell's own cellular machinery to tie off the bag. It constricts that super narrow neck of the vesicle until it just pops free. It's an incredibly precise mechanical pinch. But what if you're a naked virus without an envelope, like the coronavirus? Well, they actually use a remarkably sneaky trick. They cause cellular stress, which forces the host to create double membrane vesicles. The naked virus then hides inside this vesicle, acquiring a temporary disguise. Once hidden, it sneaks out to the cell wall and is released via exocytosis. It literally operates like a wolf in sheep's clothing to orchestrate this totally stealthy escape. All of this leaves us with one final thought. We've seen the robotic-like assembly lines, the mechanical motors packing DNA, the biological lassos pinching off envelopes. It is an absolutely stunning feat of automated manufacturing. But here is the question you should really take away from this explainer. If viruses have perfected this microscopic manufacturing and shipping process over millions of years, how can we use this mechanical understanding to engineer better wrenches to throw into their gears? Thanks so much for learning with me today, and as always, stay curious.